Hello everyone. Welcome to Biology at Click. I am Dr. Misha. Today we will discuss about NCERT Syllabus of Science Chapter 5 Life Process. By the end of this video you will learn 1. Life activities at a molecular level. 2. What is life process and why it is important. 3. Autotrophic nutrition structure and function of leaf and stomata. 4. Experiments demonstrating chlorophyll and carbon dioxide are necessary for photosynthesis. The world is broadly categorized into living and non-living things. How we would tell the difference between living and non-living things? Well, if we see a cow grazing or dog barking, our immediate perception is that these are living creatures. Even while looking at green and non-green plants, we would know that they are alive. However, when organisms are asleep, they do not show any physical movements, but we know that are living as they show physical signs of breathing. But only external movements are not signs for being alive. Certain animals like earthworm and snake can breathe without visible movements. Movements at a molecular level are constantly occurring in our body. Why we need such invisible movements to the naked eyes? Well, let us understand structural organization of an organism. The smallest unit of a body are cells. Group of cells makes up tissues. Two or more tissues types forms organs. Group of organs forms organ system. Several organ system forms organisms. However, owing to external several factors such as aging and environment, living creatures needs repair and maintenance of their structures. These are performed by several life processes. What is life process? It is a basic phenomenon that is performed by living organisms to maintain their survival on Earth. These life processes are interrelated and prerequisite for the existence of a life. Let us understand this with help of the schematic diagram. We need energy to perform day-to-day activities. This energy comes from food we eat. The food that we eat outside aids to transfer energy inside the body, and this is called as nutrition. The source of food is wide for different organisms. However, at a molecular level these sources should be broken to uniform source of energy, which are glucose, amino acids, and fatty acids. The process of respiration helps to break down complex food we eat through series of chemical reactions in the presence of oxygen to a uniform energy source. In multicellular organisms, various body parts have specialized function. Thus, food and energy produced in one part of a body must be transported to other parts of the body. There is constant release of byproducts in the body which are not only useless but also harmful. So these waste products must be removed from the body and discarded outside with the help of excretion process. Hence, all these life processes are means of existence of living organisms. In the unicellular organisms such as bacteria, the process of taking in food, exchange of gases, and excretion all occurs by one simple process of diffusion. Depending on the type of food requirement, organisms can be categorized into autotrophs, heterotrophs. Autotrophs are the self-producing organisms that uses simple food material obtained from inorganic sources in the form of carbon dioxide and water. Examples are plants. Heterotrophs are directly or indirectly depend on autotrophs for survival. We will discuss about heterotrophic nutrition in the next video. Autotrophic organisms fulfilled their nutrition requirements by photosynthesis. It is process by which organisms take in material in the form of carbon dioxide and water from outside, and in the presence of sunlight and chlorophyll produces carbohydrates, oxygen and water. Produced carbohydrates are utilized for providing energy to the plants. The carbohydrates which are not used are stored in the vacuole as starch. There is a video on plant cell structures in the cell that can help you understand about vacuoles and components of plant cells. I will link the video in the description box, please do check. Major events during photosynthesis. 1. Absorption of light energy by chlorophyll. 2. Conversion of light energy to chemical energy and splitting water molecules into oxygen and hydrogen. 3. Reduction of carbon dioxide into carbohydrates. Let's understand how each of these components are necessary for photosynthesis. This is leaf. The surface of leaf is lamina. These are veins, and center vein is midrib. The end of the stem contains vascular bundles called xylem and phloem. If you take the cross section of leaf and observe under microscope, upper layer as epidermis, the green dots on the leaves will have prominent presence of chloroplasts, and there are some guard cells at the bottom in the lower epidermis. An experiment to understand the importance of chlorophyll. Take a potted plant with variegated leaves with different colors. Keep the plant in the dark room for 3 days so that all starch is used up. Now keep the plant in sunlight for 6 hours. Pluck a leaf and mark the green areas in it on paper. Dip the leaf in boiling water for few minutes. Then immerse it in alcohol. Carefully place the leaf in the water bath and heat till alcohol begins to boil. What happens to color of the leaves? Comment down below. Now dip the leaf in iodine solution for few minutes and rinse with water. Observe the color of the leaf and compare with the tracing. Conclusion: when iodine dye combines with starch, it turns blue-black. So the green areas on leaf will turn blue while no color change can be observed on non-green part of a leaf. Let us now understand how plant obtains carbon dioxide. For that, let's understand the structure of stomata. Stomata are the tiny pores present on the surface of the leaves. Gases exchange occurs through stomata during photosynthesis. Since large amount of water can also be lost through stomata, plants close these pores when it does not perform photosynthesis. The opening and closing of stomata is a function of a guard cells. Experiment to show carbon dioxide is necessary for photosynthesis. Take two healthy plants, potted plants of same size, and place each plant on separate glass plate and place dish containing potassium hydroxide by side of one of the plants. Cover with bell jars and seal with waistline. Place the plant in sunlight for 2 hours. Pluck a leaf from each plant. Check for the presence of starch with iodine. Do both the leaves show the presence of the same amount of starch? Conclusion: potassium hydroxide will absorb all the carbon dioxide, hence, that leaf will not show any blue color change when tested with iodine for the presence of starch. That is the end of the video. Please like, share and subscribe my channel for more such lessons. Comment if you have any questions. Thank you.